growing soybeans in Missouri, wheat in the Dakotas, or oranges in Florida, the harvest is unpredictable, the elements uncontrollable.
The American farmer has always been one of our chief risk takers. All of us have benefited from that. But the farmer lives in an extremely complicated world, and that world is getting more complicated every day. It's a high-risk world. The farmer must become more efficient. The cost per unit of production must go down. The market, the international market, the demands placed by a growing population says simply that the farmer must move into a brand new age. That age is the age of biotechnology.
Over the years, science has greatly improved the efficiency and economy of farming, with chemicals to control weeds and insects, fertilizers to improve the soil, and hybrid seed resistant to disease. But improvements in the plants themselves have been achieved solely through selection and classical breeding. Breeders have painstakingly crossed the most productive, the strongest, or the most disease-resistant plants, crossing and back-crossing for generations to eliminate unwanted traits, working for many years to develop improved varieties. These classical techniques not only take time, but they are also limited to plants that are compatible with one another. Marigolds have the ability to ward off insects, and gardeners plant them around their vegetable plots to take advantage of this special ability. But breeders can't cross a marigold with a crop plant and have no way to use this natural trait. Professor Richard Meager.
Plants, which we see today, are actually separated by tens, in some cases hundreds of millions of years of evolution. Now, if we take a trait like adaptation to utilizing water, when you look at some of the plants that have adapted to arid environments, they have spines and specialized photosynthesis to protect them from water loss. Ferns, on the other hand, have adapted themselves to living in very damp environments. Here's a little aquatic fern, for example, which lives right in the water. Many ferns can stand to have their roots quite wet, where other plants would die under these conditions. Those particular properties which have adapted them to these specialized environments represent only a small percentage of the differences between these plants. And what that means to a breeder is that even primitive alfalfa may have accumulated so many genetic differences that it is not genetically competent to mate with a modern alfalfa plant.
So breeders are unable to transfer a trait like drought tolerance from one kind of plant to another. But recent discoveries in genetics, molecular biology, and biochemistry have increased our understanding of how genetic information is stored in cells, how it is passed from cell to cell, generation to generation. With this knowledge, scientists have developed the ability to transfer a gene for a specific trait from one species to another. An important part of biotechnology, this technique is called genetic engineering.
And what genetic engineering can do is focus on just a small number of genes which we would like to insert into a new plant. We're not ready to manipulate complex traits. What we can manipulate in plants right now are simple traits that require only one or a few genes. And examples of this would be something like production of a toxin against an insect, protection against other kinds of predators, herbicide resistance, drought tolerance. Genetic engineering is going to allow us to take plant breeding to a new level that it has never gone before.
Microorganisms that live in soil and air have evolved along with plants. Many have special traits which benefit the farmer. Professor Arthur Kelman.
The soil is a resting place for tremendous numbers of bacteria. When conditions are favorable, they grow with great speed. They compete with each other. So if you could hear the voices and the sounds, there's literally a war going on. But most of the organisms in the soil do not actually damage the roots of plants. And so plants have evolved in the constant presence of large numbers of different organisms, but in a happy symbiosis, you might say. One group of bacteria are able to fix nitrogen. It's applied to the seed, and it's been used in a beneficial way for many, many years.
Using genetic engineering to improve beneficial microbes could enhance their ability to protect plants from insects or perhaps from certain diseases.
One of the problems that we face today is the fact that the bacteria that cause disease in plants are very difficult to control. Citrus canker, which now requires extensive destruction of orchards and nurseries in an effort to prevent spread, cannot be effectively controlled by any of the current materials. The development of engineered organisms which could act as antagonists have potential of controlling many of these diseases which are currently not readily controlled.
Engineering plants and microbes to improve agriculture seems beneficial, but current developments have caused some public debate. A debate which closely parallels scientists' discussions at Asilomar, California in 1975 about the safety of lab experiments in genetic engineering. Professor Paul Berg was central in that discussion.
The principal concern that scientists had about recombinant DNA research was the possibility of creating organisms that might do harm, both to the scientists themselves and to the public at large. Many of us felt that it would be worth convening a conference to which scientists from all over the world could come and attend and discuss the possible dangers that might be related to such research.
The conference resulted in a recommendation that the National Institutes of Health develop a set of guidelines for laboratory research in recombinant DNA. These guidelines, overseen by the Recombinant DNA Advisory Committee to the NIH, have been followed by academic and industrial scientists ever since.
When those guidelines were established, most of the people who were involved in that procedure saw them as being temporary. It was recognized from the beginning that as we gained more experience and more experiments were done, we would learn by experience whether any of the experiments were dangerous. At the present time, literally millions of experiments have been carried out both in the United States and throughout the world, and there's not been a single instance when anybody could point to any deleterious outcome from any recombinant DNA experiment. As a consequence, the Recombinant DNA Advisory Committee has scrutinized these guidelines periodically and made them less stringent as they saw fit.
In the years since Asilomar, the engineering of plants and microorganisms has moved from theory to reality under laboratory conditions. Today, scientists have requested permission to test these products outdoors. But why is that necessary?
Well, the field testing of genetically modified organisms is an experiment. If we could carry it out in the laboratory, then we'd probably do it there first. Ultimately, to test its utility, one is going to have to test it in the open field. And in my view, if we have carefully tended and carefully protected experimental plots, then I have no qualms at all about carrying out such tests in the open.
Long before the field trial begins, other tests are done in the laboratory to determine the effect of the engineered microbes or plants on beneficial wildlife and insects. Industrial and academic scientists are devising tests for tracking, monitoring, and retrieving engineered microbes on plant roots and in soil samples during and after the field test.
The regulatory process for biotechnology and field testing is just being organized by the government.
Dr. Frank Press.
In an unusual way, the White House has taken the lead in bringing the different regulatory agencies together, the Department of Agriculture, the Environmental Protection Agency, the Department of Health and Human Services. Everybody recognizes that biotechnology is very important for the future of the country, from the standpoint of scientific research, the development of new products in agriculture and healthcare, and that it cuts across many different agency jurisdictions. And for that reason, they're proposing a method of regulation based upon the nature of the product. And at the same time, there is public concern about premature release of organisms and safety. And I think, and others agree with me, that it is possible to develop a series of regulatory procedures that are coherent and rational, that are based upon the latest scientific information, that can both protect the public, which is our primary concern, and yet make possible the release of very important new products.
Several projects have been approved for field trials, and no adverse effects have been seen. But how do we evaluate the long-term impact of biotechnology? What is the answer to the question of risk?
Risk must always be evaluated against what is the greater risk. In my opinion, there is no question in this case. The greater risk is the fear of a world with a rapidly increasing population and a decreasing resource base running out of that resource. Biotechnology is an answer to that risk. What we're talking about are making changes gene by gene in a very closely controlled way, probably more closely controlled than natural selection that comes about in nature. So risk, there's always some, but the great risk is in not using these new tools and using them immediately to guarantee that indeed we have a future.
How do scientists change the genetic makeup of living things? The secret lies in the complex molecule called DNA, deoxyribonucleic acid. DNA is present in essentially the same form in every cell of every living thing, controlling the reproduction, the growth, the health, and the form of all life. A gene is a piece of DNA, and each gene controls a specific trait. Transferring genes from one organism into another is the basis of both classical breeding and genetic engineering. The workhorse of genetic engineering is the plasmid, a free-floating ring of DNA discovered in bacteria. By applying restriction enzymes to the plasmid, scientists can cut it open at specific places. Using the same enzyme, they can cut a new gene out of the DNA of another organism. Because of their structure, the cut ends of the plasmid and the cut ends of the new gene are attracted to each other. Another special enzyme pastes the ends together, forming a new loop with a new gene firmly in place. This technique is called recombinant DNA technology, or genetic engineering. Engineered plasmids can penetrate specially treated bacteria, like this agrobacterium.
Agrobacterium tumefaciens makes its living using genetic engineering. It does this by inserting a small piece of its own TI plasmid DNA into the plant DNA. Those genes which Agrobacterium uses to take over the metabolism of a plant and to cause it to form a gall or a tumor have been removed from these disarmed strains of Agrobacterium. What we have done as genetic engineers is to insert a gene into this region, and the Agrobacterium will do its natural job of genetic engineering for us, transferring any gene of interest into a broadleaf plant.
The agrobacteria are grown on culture dishes. After colonies have formed, those that have received the engineered plasmid are transferred to a test tube where they reproduce themselves in large quantity. The engineered agrobacteria are then poured on special culture dishes, and pieces of plant leaf are added. Inside this piece of leaf, at the level of the single cell, the transfer happens. Cutting has wounded the cells on the edge of the leaf piece. The engineered agrobacterium binds to the cell and duplicates the engineered plasmid it carries. Wounding has caused a chemical signal to be sent from the plant cell to the bacterium, which enables the plasmid to transfer the new gene into the plant cell. The new gene travels into the nucleus, where it becomes a permanent part of the plant cell chromosome. When the plant cell divides, each of its daughter cells will receive the new gene. Unlike higher life forms, whole plants can be regenerated from single engineered cells using a technique called tissue culture. The leaf pieces are removed from the agrobacterium solution and placed on media which contains hormones and nutrients to encourage growth. After a period of time, callus, undifferentiated tissue, begins to form. Then shoots emerge from the callus and roots begin to grow. The engineered plantlets are transferred to soil, grown to maturity, pollinated, and their seeds harvested to grow into a second generation, also containing the new trait. This is the process Professor Roger Beachy used to give plants a new level of resistance to viral disease, which costs farmers as much as $200 million every year.
Our approach to creating virus-resistant plants has been to take a small piece of the virus genetic information and to insert that small piece of information into the chromosome of a plant and to produce from that cell a whole plant, and in so doing, the plant has become resistant to an infection by the virus that was used initially. When these tomato plants then are infected with tomato mosaic virus, those plants that are not resistant develop normal symptoms of the disease, chlorosis and stunting and generally sick looking, whereas those plants that are protected by the gene transfer mechanism grow very normally and healthy as if they had never been infected with the virus. Traditional plant breeding approaches take between 5 and 7 years for a plant breeder to produce a virus-resistant crop species. What we've been able to do by the new direct gene transfer approach is to produce disease resistance within 9 to 12 months. The farmer has learned to live with between 5 and 30% crop loss each year caused by virus infections. Bringing in new sources of disease resistance, it should make it possible for him to reduce that loss to either nothing or a much smaller level.
But agrobacteria only work in broadleaf plants, tomatoes or soybeans.
For over a decade now, people have been looking for natural genetic engineering systems which can be used on the grasses, particularly the cereals like wheat and rye and barley and corn. There are a number of viruses which already know how to infect these particular groups of plants. And one of the plans of molecular biology today is to try to characterize these viruses well enough that we would know how to remove their pathogenic properties, use them to carry a foreign gene and spread it through a plant, but not cause any of the symptoms which it normally causes. I don't think there's any question that within the next few years we'll have some virus genetic engineering systems which are safe to use in cereal plants.
Engineering systems that work on wheat or corn could be used to improve the nutritional quality of the seed itself. Professor Brian Larkins.
Most seeds contain a fair amount of protein. In cereals, it's about 10% of the dry weight. About half of that protein is what's called storage protein. What we're trying to do is to modify the genes that encode those proteins so that they will have a higher lysine content. Now, normally in the diet you need about 3 or 4% lysine, and with corn you only get about 1% lysine. So if we could increase that percentage up to about 3 or 4%, then corn protein would have the nutritional quality of milk protein. Often these vegetable proteins are a principal source of protein in third world countries such as South America and Africa. So if we could improve the nutritional value of these proteins, we could provide in a single package almost a far more balanced diet for people in those countries.
Frost damage costs farmers and growers between $1 and $15 billion every year. Some of this damage is caused by bacteria that help ice to form on plant leaves. Professor Stephen Lindau has used genetic engineering to delete the bacteria's frost-causing capability.
Ice-nucleating bacteria were discovered about 10 years ago at the University of Wisconsin as part of my work and others. And it's now known that virtually all plants, especially in temperate areas of the world, are covered with these bacterium. About 1% of all microorganisms on plants are of a species that can cause ice formation. Well, about 5 years ago, Dr. Cindy Orser, a student of myself and Dr. Nick Panopoulos, was first able to clone the genes that confer ice nucleation in Pseudomonas syringae and Erwinia herbicola, the two most important bacteria that cause ice formation. And with the gene physically in hand, it was rather simple to construct deletions within the gene that no longer allowed it to function.
When plants are coated with bacteria that no longer contain the ice nucleation gene, they are able to withstand temperatures which otherwise would harm them, at least in the laboratory environment.
There are a lot of variable biological and environmental factors which are very difficult to reproduce in the laboratory. And to know, and really the basis of our trial is to study how this strain interacts with all possible outside organisms. We've done a large number of interactions in the laboratory, but we can't always obviously do all of them. So to study these large numbers of interactions of microorganisms and see how they deal with various environmental factors is really the goal of our study, and really can't be done in any reproducible way in the greenhouse. We have very sensitive ways to measure the presence and activity of these microorganisms in nature. In this case, we have very specific media on which these microorganisms can grow. Certainly we have to be aware of the potential misuse of this and the safety of handling this tool, but biotechnology is a very important tool. I'm very concerned that we have this tool available to better study biology. It's nothing more than a very powerful microscope was 100 years ago. It's a new way of looking at biology.
Another microbe, called Bt, naturally protects plants from insect attack. Professor Kurt Thorne has studied Bt for more than 15 years.
Bt, or Bacillus thuringiensis, is a soil bacterium. It's a spore former. It produces a substance which kills insects and has been used commercially for years to combat insect pests. It produces a protein which occurs as a crystal. The protein itself is not toxic. It's called a protoxin. And the larvae of the insects ingest the protoxin, and in the gut of the larvae, it is digested and produces a toxin, which then kills the insect. Since it's not toxic to any animals, it's used in the home garden. There are no harmful effects.
Biological control of insects, taken from nature. Control which could be made more effective through biotechnology.
If you could clone out the genes that are responsible for toxin production and put them directly into a plant, then the insects, as they eat the plant, would come in direct contact with the toxin. It could be much more effective than it is in Bt itself.
Asia has made fantastic progress over the past 25 years in its capacity to feed itself.
Dr. Niall Brady.
Even though there still are more hungry people in Asia than any other part of the world, they have used science and technology, and they've used policies that made it profitable for those technologies. In Africa, the problems are very severe, and as a consequence, Africa's capacity to feed itself is going down. I see no way that the Africans are going to be able to feed themselves unless they have technologies that fit their conditions. We believe that through biotechnology, it will be possible to develop plants that would tolerate those high salt conditions, those high acid conditions, those conditions of high aluminum toxicity. It would make it possible for farmers to grow good crops in areas where they're simply not able to do so at the present time.
reduce risk, to increase efficiency, to begin to control the effects of weather and the elements, to help the world feed itself. This is the goal and the promise of the new biology for agriculture, the promise of the earth.
I believe that biotechnology presents to us today opportunities that are so great personally that we can't conceive what those benefits will be. We only begin to see some of the indications. Those plants that can produce their own pesticides, those plants that are drought resistant, those plants that indeed could be even irrigated with brackish or salt water, those plants that can take the farmer, the producer, to the point that he would have multiple markets for that produce. It may be in the productive economy. It may be in the human food chain. We are on the edge of a brand new day. It's a little frightening to some of us, but the fact is we're there and our very survival depends on it. It can be the most exciting period in my estimation in recorded history. I think personally it will be.